Hi everyone, welcome to workshop 11. We're going to be continuing on about digestive. Maya and I are going to cover 47.3. First, let's start off with a quick overview of this section. We're going to go off first with talking about dietary needs, types of minerals and vitamins, the amino acids and fatty acids that are required and that you receive energy from. And then from there, we're going to go through each part of the digestive system, so the function of each organ and the whole digestive tract. And from there, we're also going to be going over the role and breaking down and the absorption of food. Important key things to recall for this chapter is the basic knowledge of amino acids, proteins, lipids, nucleotides from Bio 1, because since we are going over like nutrient absorption, there's a lot of terminology from Bio 1. But overall, I think this section is pretty straightforward, and the processes of the digestive system is overall pretty simple to understand.
When talking about amino acids and fatty acids, there's 9 essential amino acids for the human body's function. So lysine, histidine, tryptophan, phenylalanine, threonine, valine, methionine, leucine, and isoleucine. So if you took Bio 1 already, you know that there's 21 most common amino acids in the body. So these ones are they're essential for humans, and these are usually supplied by proteins in animals. So in like a regular healthy diet and with some protein, you can receive these amino acids. So they're required to synthesize enzymes and other proteins. And if there's not, so for example, like a plant-based diet, the proteins, these kinds of amino acids are not complete in those kind of diets. So they don't provide a sufficient amount of each one. And if a diet lacks then these amino acids, many enzymes and other proteins can't be synthesized in the sufficient quantity that they're needed. So going on to the 2 essential fatty acids, it's linoleic acid and linolenic acid. So they're required to synthesize phospholipids that are used to form parts of like some hormones and then biological molecules. These are easy to obtain fatty acids in a regular diet, so most people won't have a problem obtaining them.
Vitamins can be categorized into 2 types. So there's hydrophobic and hydrophilic. Hydrophobic meaning they're fat soluble, and kind of you can think of it as like water hating. And then hydrophilic is what you would call water soluble, so water loving. And examples of hydrophobic vitamins are vitamin A, D, E, and K, and they're usually stored in if there's excess of it in adipose tissue. So if a diet is lacking in, say, vitamin A, D, or E, the body goes to those excess in the adipose tissue and they can be easily used. And so for hydrophilic, on the other hand, if there's excess of it, the body secretes it through urine because it's soluble through water. Going on to the types of minerals, some macro minerals that are usually consumed in like ion form are calcium, chloride, magnesium, phosphorus, potassium, sodium, and sulfur. In your book, there's like a nice table of all the different types of minerals. So for your macro minerals and then trace minerals, I don't think you need to know too much on this, but it's a nice resource to look at about the functions of the minerals and if there's a deficiency in those minerals and how is it's going to affect your body. Usually, if a diet is lacking in these minerals, there are supplements that can be taken. Additionally, if there's an excess intake of minerals, it also can affect your body in a negative way. So say if there's too much iron being ingested, it can be linked to liver and heart and blood vessel damage and too much sodium can also lead to elevated blood pressure. And at the end of this slide, you can just see the kind of embodiment, the body's requirement of minerals per day. Now we're going to go over the layers of the digestive tract. These layers are going to be consistent and they're going to be in every part of the digestive tract. So when you're from entering into the mouth and then down into like the stomach and small intestine, there's going to be these 4 layers, but it might differ in function or in an added layer depending on where in the digestive process the nutrients are being consumed or like absorbed or digested. Going over first the mucosa, which is the most inner layer, which is made up of epithelial and glandular cells. This layer is what's being interacting with the content of like the nutrition content, and the epithelial cells are helping with the absorption and the digestion of the nutrients, while glandular cells are involved more with secretion of enzymes. Next, in the submucosa, it's a thick layer of connective tissue, and it's containing like neural networks from the enteric system, which is the if you remember from the nervous system, the enteric system is the one that's kind of controlling the digestive tract. In the submucosa, there's also blood vessels and a lymph network. The network of blood vessels are obviously going to be supplying nutrients and oxygen to the digestive system, and then along with the blood vessels, there's also the layer of lymph networks. Going over to the next layer, which is the muscularis, it's kind of sounding from the name. It's the layer where the layer of smooth muscle is going to be. So there's 2 types of smooth muscle in this layer. There's circular and longitudinal, and they coordinate together to move the contents of that's being digested. So from one organ to the next, these muscles are going to be constricting and pushing the digestive contents to lead into another organ and or part of the digestive tract. And lastly, the serosa, which is the outermost layer of the digestive tract. It is made up of connective tissue that secrete a fluid, and that fluid's function is basically to lubricate the area between organs and the muscles on top of the digestive tract, and it's aiming to reduce friction between like muscle movement and allowing the organs to move as they need to. And on the slide, I just have like a nice little diagram of how these layers look and from where they move from. So in the innermost layer, you can see the mucosa, and then the submucosa, and then the muscularis and the serosa. From this diagram, you don't need to know like the specific terms that are mentioned here, like the lamina and propria. It's not required for this class. I just thought this was a good image I found, which really shows the layers nicely.
Digestion begins when food enters the mouth and is ground into small pieces to form a bolus. As food is chewed, it mixes with saliva. This acts to moisten the food for digestion. Bicarbonate ions in saliva maintain a pH of 6.5 to 7.5, the optimal pH for the enzyme salivary amylase to hydrolyze starch to maltose. Saliva also contains lysozyme to kill bacteria. When food has been thoroughly chewed, the tongue will push the bolus to the back of the throat, where it will activate touch receptors. This triggers the swallowing reflex pictured to the right. Initiating this reflex can be under voluntary control, but once the reflex begins, it is under autonomic control. In this reflex, the larynx moves upward, so the epiglottis is now blocking entry to the airway, while the pharyngoesophageal sphincter opens to allow the bolus to pass. The bolus is actively pushed through the esophagus by peristalsis, a coordinated contraction of circular muscles. When the bolus reaches the end of the esophagus, the gastroesophageal sphincter opens to allow entry to the stomach. In certain individuals, this sphincter doesn't close completely, and stomach contents can enter the esophagus in a condition known as acid reflux. Food entering the stomach activates stretch receptors that stimulate secretion of gastric juices from the mucosal layer of the stomach. This layer is covered in gastric pits, inside of which are gastric glands containing parietal cells and chief cells. Chief cells secrete pepsinogen, the precursor of pepsin, a type of enzyme called a protease that cleaves proteins. Parietal cells secrete hydrochloric acid, which performs a number of functions. Hydrochloric acid cleaves pepsinogen to form pepsin, while simultaneously lowering the pH of the stomach to 2. This is optimal for pepsin activity. This acidity also acts to denature proteins and kill most bacteria. However, it does not kill Helicobacter pylori, or H. pylori, the bacteria responsible for peptic ulcers. To protect the stomach lining from degradation, inside of gastric pits are also mucus cells that secrete a thick coating of alkaline mucus. After digestion, the food is now called chyme and is pushed past the pyloric sphincter into the duodenum of the small intestine. The chyme enters the small intestine, joined by secretions from the pancreas and liver. Both organs secrete bicarbonate ions in an effort to neutralize the acidic chyme. The pancreas secretes a number of important enzymes, such as pancreatic amylase, proteases trypsin, chymotrypsin, and carboxypeptidase, pancreatic nuclease, and lipase, an enzyme that breaks down fats. The liver primarily secretes bile, a mixture of bile salts, cholesterol, and bilirubin. Bile is stored in the gallbladder until chyme enters the small intestine. Bile salts are derivatives of cholesterol and amino acids. Like soap, bile salts have a nonpolar region and a polar region, which allows it to act as an emulsifier. This means that the hydrophobic fats are no longer clumped in large globules, but mixed with the aqueous environment of the small intestine, allowing for greater digestion. The lining of the small intestine maximizes surface area by folding into ridges covered in villi. These villi are lined with brush border cells that have their own microvilli. While digestion of fats occurs in the lumen of the small intestine, these brush border cells contain enzymes disaccharidases and aminopeptidases that complete digestion of proteins and carbohydrates. Here are some diagrams to help you better visualize the topics discussed on the previous slide. On the right, you can see the organization of the intestinal lining, starting with a slice of the small intestine. The hollow area in the center is the lumen, surrounded by the folds and ridges of the lining. These ridges are covered in villi, shown in D, as well as the larger picture on the left. The villi are lined with brush border cells that have microvilli on their apical surface and contain enzymes, aminopeptidases, and disaccharidases. Sugars and amino acids then diffuse into the capillaries. Fats, digested in the lumen, are absorbed by the lacteal and are now in the lymphatic system. The capillaries of these villi collect into a larger hepatic portal vein. The hepatic portal vein brings blood from the digestive tract's capillaries to the liver's capillary beds. The liver contains many enzymes that chemically process nutrients. One example of this is glycogen synthase, which pulls excess glucose from this nutrient-rich blood to make glycogen. When blood glucose levels are low, glycogen phosphorylase, another liver enzyme, breaks down glycogen. Another important example of this is the activity of cytochrome P450 enzymes to metabolize potentially harmful chemicals. In the pharmaceutical world, this is known as first-pass metabolism. Drugs administered orally must be taken in a much higher dose than what would otherwise be necessary because only a fraction of the drug will make it past the liver. Finally, food leaves the small intestine and enters the cecum of the large intestine, right next to the appendix. The large intestine is relatively smooth, as its role is to absorb the remaining water and ions and pack the indigestible material into feces. Over 1,000 different species of bacteria live in the average human colon, feeding off the remaining nutrients and producing fatty acids and vitamins K and B vitamins, which can be absorbed by the colon. The most common of these bacteria is E. coli. When the feces reach the end of the large intestine at the rectum, stretch receptors trigger the defecation reflex that opens the anal sphincter. Digestion of the food has been completed. That wraps up this part of the chapter. This table provides an overview of digestion and the enzymes involved at each part of the digestive tract. Please refer to your book and the slides for any extra details not included. And as always, if you have any questions, feel free to reach out to any of the TAs or Dr. Wilson for clarification. Thanks for watching, and I hope you are all staying healthy. Have a great rest of the semester.